So go ahead, Terry. Okay. I'd like to start by acknowledging my co-authors, Rowe, who is my genetics lab manager, Gardner, who's over here, and will be giving another talk shortly, and Jim Long, who has been a mentor to many of us at Utah State University in Aspen and silviculture. Also, MaryLou Fairweather, who is a collaborator down in Flagstaff, Arizona. So, as you've heard before here, aspen has two basic means of reproduction. So should do that. One is vegetative through suckering, and the other is sexual or through suckering in aspen is under hormonal control. So if you have very large stems that are connected to the root system, they can suppress suckering in the roots. And once you take those large stems out, either by coppicing or by fire, however they suffer mortality, the suckers are then released from the roots, sometimes in astounding numbers and densities, which you can see right there. Suckering over time can lead to the establishment of really large clones on the landscape, and this is a very dramatic picture of that. I looked hard to find this one. But, it can lead to these clones are single individuals that ultimately came from. So we've got multiple trees, multiple stems, sometimes called ramets, but they are all the progeny of a single seed. Aspen also produces plenty of seed, and as we heard Bob mention before, these seeds have high viability rates, but they're rather picky about where they can germinate and what can lead to successful germination. Seedling establishment seems to require mineral soils, and it seems to require moisture in just the right balance. Because we don't seem to see seedling events very often in western landscapes, and I should clarify, by western landscapes, I have a terrible bias here. What I really mean is western U.S. Sorry, Alberta people. But in western U.S. landscapes, you don't see these seedlings very often, or that at least you don't see these seedlings very often. And it's thought is because of the environmental conditions that prevent seedling establishment. So that's. I would like to mention, though, that this failure to may actually be a detection problem. If you can imagine a very dense patch of suckers, the ability to detect seedlings is awfully low. In addition, you might not be able to detect vegetation backgrounds, or if they're kind of scattered and not real patchy with lines. Contrast, and we can dismiss them. So based on this understanding of reproductive biology in aspen, this is sort of the traditional in place for some time of reproduction in western aspen. First off, the idea is that aspen exists primarily in large, ancient, and clones. By cohesive, they're all in a blob. They're not just the same clone. It's also thought that stands generally represent one or just a small handful of clones. It's also thought that most clones were established from seed a long time ago, and by a long time I mean Pleistocene. Remember, I'm talking about the traditional view. And also that aspen is maintained in landscapes entirely by suckering in the western U.S. That sexual reproduction, and that the loss corollary here is the loss of aspen from western landscapes, unless you can't spread out into it. However, our lab and others have come across some patterns in aspen and western landscapes that are challenging. challenging a lot of these traditional. And one of the reasons that we're suddenly coming up with these challenges is because we've got a new toolbox. And my lab is a genetics lab. We're using genetics tools to find clones. Past, before the advent of some of these genetic tools, we were looking at leaf morphology or phenology or what it was to define clonal boundaries. And again, because of that human tendency to pick out lines, separating clones. That's where our traditional view, I think, came from. Anyway, we now have this new system. For the studies that I'm going to be describing today, we're using microsatellites, which are a rapidly mutating kind of marker. They're appropriate for individualized forensic crime scene, for example, or jobs. So these are the kinds of by genetic individuals. Aren't you glad that's all I'm going to say about genetics? You know, let's go back. I am going to jump directly to some of our results without talking too much about the methodology. One of our first clues that something else might be going on in terms of clonal diversity came from a landscape called Swan Flats, which is off of Logan Canyon, and we didn't have in mind to take down this traditional view. I thought it might be interesting to see. And we're told by the foresters and people that knew that we should expect maybe 10 or 12 clones. So off we went, we sampled, we wanted to find what boundaries were, so we sampled 812 stems on a 50-meter grid and detected 190 Rowe. So that was a surprise to us. We did find that these clones were generally cohesive for the most part. Most clones, however, were represented by 1 to 2 stems of the food. Only 22 of these 195 clones were represented by more than 10 stems. In this slide, we've used colors to denote the clones out there, different individuals. But with 195, we ran out of colors in a big hurry. And so I just used black dots to denote clones where we only saw black dots here have a different. Here's a little bit of a close-up. I think the big message here, the first one that we came across, was that stands are not equal to clones. In fact, the idea that clones, that stands are represented by only one or even this is a distribution of the, you can think of this as a clone size. So on the x-axis here we have clones. On the y-axis it's the number of stems sampled. And you can see lots of stems. By and large, that was not the story. So, this led us into the field of self-doubt. We began to wonder, gosh, is this something we did? And the first question that came to mind was about something called somatic mutations. Somatic mutations are genetic changes that happen during the lifetime of an organism, like a freckle on a human. That can be a mutation that shows up during the lifetime, but it's not something you pass on to offspring. So we worried that maybe with these fast mutating microsatellites, that maybe what we were really seeing is variation with somatic mutations, and in fact the clones were really big, but well, we were dividing them by counting every somatic mutation as a different story. We thought about that a lot. And we decided that we better grant. And we did by using something called a mismatch distribution. And a mismatch distribution is just a way to look at the genetic mismatches between two clones. So if you have a somatic somatic mutation might have a clone that has a certain makeup, but it has one marker that's different from the rest, right? And that's what you would expect with a somatic mutation. If you have, however, two clones that really came from two different you expect a lot of differences. Markers. So this is kind of a cartoon diagram. This is not real data, but showing that if you have somatic mutations or lab or scoring errors, you expect just a few mismatches between genets and a lot of molecular markers. On the other hand, if you're really looking at genets that are truly different, you expect a distribution like this with lots of mismatches. This is real data, and it was a real signal, and this is the distribution of mismatches from Swan Flats. And I had a 0 category, but you can see that our low category, where you expect that mutations or lab error, negligible compared to a little further evidence. This is reaching way back into genetics. More genetic evidence that the populations were in higher Weinberg equilibrium, like any other population that you might look at. Also, also disequilibrium. I won't bother to explain that, but if you know what it is, you'll be happy to see that. So overall, this means that different genotypes are most likely produced by sexual recombination, not accumulated. So in summary, at Swan Flats, many clones, most of them very small, and the distribution of clonal richness, right, the number of clones per unit area, was very patchy. That's exactly what you would expect out of sexual reproduction. You expect to see that seedling is patchy. You expect the clones to be very small at first, and they expect out competing each other. Then, more self-doubt. Well, if there's nothing wrong with our markers, maybe there's something wrong with our site. About the site. Oops, if you look at the distribution here, this really, to me, suggests a seeding event. Here, the white dots, we ran out of symbols again. The white dots here, 4 stems. Another interesting pattern, and I'm not going to say much more about this today, but in both study sites, the big that was kind of a surprise, including Pando, by the way. Triploidy means more vigorous vegetative growth. It also means you expect them to be sterile. Our lab's working hard right now on the rest of this story. That'll be our next chapter. Okay, still though, what? Is this really the story? If we're seeing all this genetic signature of seeding events, given the small size of these clones, why do we see more seedlings on the landscape? That's kind of a mystery. Well, you start looking through the literature, and actually, people do see seedlings. And this is just sort of a sampling. In Bob's of the literature, talked about other people that have. They do have, and they just didn't make it into the aspen. More, I guess. So we do seem to find aspen seedlings. They tend to be in an area. How much of that is because there you have it. In the rest of the time, I'd like to talk about 2 more recent documented. One is a pumpkin fire, which you may have heard about. And you can see it in the background here. Burnt ponderosa. in an attempt to establish ponderosa trouble with it, and so they put up these exclosures to establish ponderosa pine plantations in areas where there was aspen before the fire. And we have good documentation of that. And up came the ponderosa, but up came the aspen too. So the seedlings that are established in these protected plantations, I thought was really interesting. MaryLou Fairweather was the one to notice this, and she thought, well, how can this be? And we wanted aspen seedlings. She sent samples to the lab, and we were able to based on 49 samples. Indeed, every one of them. That's clearly a seeding event. Then, I should mention this too. We did find some aspen established outside of this exposure. But if you look closely at this picture, you probably see a reason. Outside of the exposure of ponderosa, and so we did see them coming up. The message here is that it's aspen seeds. In 2010, another big fire in Arizona, the Schultz fire. In 2011, who was very prone went up to the side of the Schultz fire. This was just this past summer, and what she lots of aspen seedlings coming. When she first described this to me, she said, like a carpet of aspen. She revised it later, and she said, well, it's kind of a thin carpet. But this was covering a big area. And we worked on this to establish that there are seedlings. This was an easy way. And a lot of sure enough, they are not connected to anything at all. So this is yet another seeding event that occurred in Arizona. So in summary, we're finding that most in these landscapes, and I'm not showing you other data, but we have data from a variety of western sites as well. And it looks like most clones are small, which is not what we were expecting. That recent sexual reproduction is likely. It explains both observation of seedlings, but also that sexual reproduction is certainly not negligible. That recent sexual reproduction seems to explain a lot of the aspen on the landscape, the land of Pando. That stands usually contain many clones, and clonal richness is patchy. In terms of I would say that the identification and protection of recent seedling events suckering establish those clones, but this could be hugely important. For aspen to adapt, because sexual reproduction is how we get the genetic substrate for natural selection, act and adapt to further. Okay. In addition, aspen seeds are an important part of aspen restoration programs. I'm not thinking about aspen this way. But for example, has anybody heard of leaving seeds for aspen? But perhaps it should be. In addition, if we can figure out how effectively to establish aspen from seeds, reestablish aspen in new areas. Right now, if we just count on coppicing and suckering. Okay, so I would like to acknowledge the funding that we've gotten. A few seed grants, really from USU, and a bit from the USU, and a lot of graduate students. I would have. I'm sure I've left off lots of names here. We had an army of people out there collecting aspen leaves